then it may be difficult because if I go to full screen, it may hinder. Yeah, let's see how it goes. Can you see my full screen? Yeah, and can you also see me go through my slides? Yeah, good. Yes. Thank you. Well, I'm going to try to be very quickly. I try to be not longer than 20 minutes. It's an old presentation. Here you see Roland with his back to the camera with a group of students of the Biohack Academy, but also students of the Art School of The Hague, making competent cells, filmed by the VPRO, which is the Protestant television channel in the Netherlands. This is always interested in provocative television. And they think they're going to do some CRISPR mutations here. However, we go as far as making competent cells, which is something you also do in CRISPR mutations, and then give them the ethical question whether in the heat shock they're going to apply a CRISPR mutation or not, and by doing so, violating legal rules, because we are not a certified lab that is allowed to really do CRISPR. We're housed here. We have a number of labs of the Waag. One of the spaces of the building you just saw has this picture. That's the anatomical lesson of Dr. Tulp, painted by Rembrandt van Rijn, which is a very early art-science interaction that happened in this very building. Yeah, that gives a lot of meaning to what we do here, the dissection practice of why CRISPRing artists are interesting. This is a little bit what we do. I'm going to skip these because of time. So quickly, what is going on? There's quite some very provocative things, and these are even older images that is going on. We know that there is full mammalian animals made in extrauteral wombs in plastic containers, like you see below left at Tokyo University. Organs are printed by Organova, however, they're not that successful, but it's also very interesting to know that you can print DNA and also observe the sequence of DNA by dragging it through a protein screen that you see below right. It's interesting to see reactions of biohackers that make their own PCR machines, and above right you see the blacklist printer of Georg Tremmel in Tokyo. That is supposed to print pieces of DNA that you are not to have synthesized, because they are pathogens that only big commercial companies are allowed to make for you under certain conditions. So it's interesting also to see that there is artists working with very complex biotechnologies as a reaction to those laboratories being opened to them. So we welcome these artists at the Open Wetlab in the Waag. And here you see a picture where Adam Zaretsky and Oron Katz organized a workshop where you could be participating in making cell cultures of meat. So actually, yeah, mimicking meat or making meat grow in a petri dish, which is something that in the disembodied cuisine project of Symbiotica, yeah, was discussed, invented, and it's interesting to see that commercial research is is taking that over and really offering it as an alternative. So that's a little bit the reverse that is happening. It's not artists reacting on novel technologies, but novel technologies reacting on artistic practices. We done so again once in a participatory workshop at the Waag, organized by a designer, Chloe Rutseveld, but also during that event discussed the provenance of the idea of making laboratory meat. Because we used to eat offal, we used to eat other animals, and if you look in a supermarket and you see those square cubes of minced meat, it almost looks like laboratory meat already. So we had the group that is called the Cuisine of the Unwanted Animal cook for us. And I cooked with them a mouse liver parfait of the mice that you see lower right. But we also cooked muskrats that are caught by the thousands in the Netherlands because they dig into our dikes as an invasive species. So the Cuisine of the Unwanted Animal cooks a lot with invasive species. I think it's also interesting to say something about these type of art practices that also ask attention for research that is done, but is not so much made public, about endocrine disruption chemicals that you find in surface water. Mary Magic is specialized that a little bit in that a little bit, and she's doing a lot of experiments looking at how these types of xenoestrogens she finds that leak off plastics that are left in nature change the forms of the bodies of microorganisms and therefore disrupt their natural reproduction, even doing so with larger mammals. Here you see a course again that is do-it-yourself hormone extraction that was more or less guided by Mary, but also guided by Spela Petric at the Waag Lab a few years ago. This is a map that I often use, a map made by Adam Zaretsky that shows you the diversity of vivo arts, but also shows you a kind of diagram that leads to these transgenic hereditary biology arts that he has a high interest in. So then these types of art that use gene editing or genetic modified organisms as art, this is regarded one of the earliest ones of photographer Edward Steichen, who used radiation to create another type of delphinium's blue that supposedly are still buyable on the seed market for flowers. It's interesting that he does it with flowers because the flower industry is also a big industry in the Netherlands, and it is an aesthetic type of market that is not art, but in its aestheticism, is very similar to art. The very early other one, or the very early other one, I don't know whether it's a very early other one, but another well-known one is one that Gunther also mentioned yesterday, Micro-Venus of Joe Davis, where, correct me if I'm wrong, the more conventional type of viral transfection was used to code an image that represents the female reproduction organ into an E. coli bacterium that he wanted to have in a spacecraft and in a module in that spacecraft that could be unpacked by intelligence life somewhere else in the universe. Might it ever get that far? Very well other known piece is ALBA of Eduardo Koch, and also his piece Genesis. And Genesis is a piece of text of the Bible that was encoded also into the bacterium. Since we have the pleasure and the honor of having Marta de Menezes here, I had also this image in my presentation, yet I have to admit, Marta, and maybe you can say something about it yourself, I don't know the exact technique that you used here to alter the butterfly wings. Could you tell us?
Sure. Thank you for bringing this up. I can talk a little bit about, so the first thing you need to know is that this is not a genetic manipulation. Even this week, I was contacted by a production company asking me how I genetically manipulated the butterflies in 1999. And the first thing I had to tell them was, well, actually, they're not genetically modified, which sort of shocks everybody. It's still puzzling to me how they misread everything that has been written about these butterflies. So this is actually a very old technique in terms of maybe biological techniques. It was something that was discovered and taken upon since the 60s by a very famous butterfly scientist, Nijout, where you do microcautery. And what happens in microcautery is that you kill a few cells in the butterfly wing while they're forming the pattern, and this interferes with the signal between the chemical signals between the cells. So actually the cells in the center of the eye spot, for instance, are responsible for the whole formation of the eye spot. So if you, at a specific time of the development, kill those specific cells, the eye spot is not occurring. Or in the case of this butterfly that you here see, the brown one, if you interrupt the transmission of that signal around those central cells, then you can delete parts of the eye spot as well. It's a mechanical technology. It's an effect caused by inserting a very thin needle into the pupa of the butterfly very, very lightly at the right time of the development of the butterfly when it's inside the pupa. And it creates this extraordinary effect, which only is visible through your eyes. So if you look into the genes of these butterflies, they're exactly the same. If you see under the microscope, there are no scars because it's done at a developmental stage that killing a few cells is not going to interfere in terms of smoothness of the surface cells. And there's no clear effect in terms of behavior or in terms of there's no effect in terms of lifespan. So the technology is a mechanical one. The knowledge is based on evolutionary and developmental information, and the butterflies are just the clue, or the visual clue that the wing pattern is different is the only clue that you have that these are not natural butterflies. The offspring reverts back into wild type pattern.
Yeah, so it's a non-hereditary change.
Yeah.
Yeah, yeah. As where a lot of the others, yeah, in some cases lead to hereditary changes. The super wheat kit of Heath Bunting, actually, he told me about making it that he was making it in '98, and actually for me, personally, this was the reason that I got interested in this biologically oriented type of media arts, bioarts. Also because it's because of its really activist attitude. It's a modified wheat in that he is proposing as an artwork that you could really have in order to fight the monoculture crops of corporate genetically modified crops, in fact. Here you see, and that is also a type of similar viral transfection of Margarita Pevere, where she is doing a story that she recorded from an elderly lady in Germany that she put in the code of this kombucha type of organism. And she didn't disclose the exact story that she put in there. The technique is a viral transfection, which is kind of conventional already, because there is many artworks known to use this type of technique. Also this one, if I'm not wrong, this is also a viral transfection, where an artist was able to use some material, and I think it's a bit of hair of Elvis that he put in the genetic information of a mouse. Yeah. So is this Elvis? Is this Elvis #1 or #2? Yeah. I think the kind of humor here is very interesting. Another type of art using genetic modification here is bulletproof skin of Jalila Saidi. She was using spider silk that was produced in the milk of a transgenic goat, that she had silk weavers make a number of patches of, because the goat can produce larger quantities of the silk thread. And if you have a 8-layered patch and you let that grow in a cell culture of human skin cells, in this case, it would hold a bullet shot at a velocity of 329 meters per second. I was with the experiment on the shooting range, and of course, it will harm your body, but it's interesting that she creates this type of narrative that is about the trust in these materials, that they may take these type of violence, and people believing in that you should be using or developing these kinds of technologies to protect yourself. I wonder whether protection really happens if you create these materials. But it was a very interesting and complex kind of research that she did here. Blighted by Canning, Charlotte Jarvis, who also used viral transfection to put the first article of the Declaration of Human Rights into a type of fungus that is infecting apples. She wanted to send the apples around to other labs to be decoded there, but she was not permitted. That was one of the first requests for permission we did in the Netherlands to send those apples around. That was denied by the authorities to happen. Thereafter, we had This was the third one. So that was not the second. This was the, oh no, yeah, this one was the third one. This is Howard Boland showing the way he is looking at changing the color of bacteria as a type of artistic visualization and using transgenic techniques for that. But on the right side, you see him doing all sorts of tests with magnetic bacteria that were also genetically altered. We also asked for permission for that work to be shown in an exhibition, which was also denied by the authorities. Then going back to this work, the Aerorarium of Adam Zaretsky, where he used zinc fingering technique, which was a technique just before CRISPR, a very complex one, to alter the morphology of small Arabidopsis plants in such a way that you wouldn't get one new type of feature, but an enormous mix of new features, so that it becomes scientifically irrelevant to research, which was what he called the errorarium. It had so many errors, which is a kind of a critique on scientific reductionism by him. But it's again done in the Sylvius lab in Leiden, but it was not allowed to leave the laboratory. So the work of Charlotte and that of Howard Boland and that of Adam Zaretsky were three types of genetic modified artworks, let's say, that we asked the authorities for permission for exhibition. That was denied, but it led to a discussion within that same the same institutions that did not give us the permission. And in the end, they changed the law a little bit so that also non-certified, non-academic labs were allowed to have a permit to do certain types of genetic modification, however, on a very narrow bandwidth. Yes, here on the right side, you see a work that was granted the Science, Technology and Arts Prize of the European Commission. I Am Humanity, which is actually a song that was, as such, again, using viral transfection put into an E. coli bacterium. What I don't like so much is that the whole image you see here is not a real image because the green, yeah, I wonder whether the green is so real there. Another Starts Prize honorary mention was given to Georg Tremmel, again, for his blacklist printer. Anna Dumitriou is another artist that also works a lot with genetic modification and arts. And here you see a work that's called Make, Do, Mend, or Controlled Commodity. And it's very much about the story behind the work. The dyeing industry is an industry that actually was a precursor for the pharmaceutical industry. A lot of the chemicals developed there also were tested as medical chemicals, among which was also antibiotics. She is researching a lot the history of tuberculosis, and in 1941 the first antibiotics in England was tested on humans, but it was also the year in which the so-called controlled commodity campaign of the UK government was started, that was motivating all citizens of the UK because of the war to reuse old clothes, so repair them. Yeah, reuse them. Be careful with all your commodities. And Anna used that idea of controlled commodity to make a patch that you see upper left that is colored by bacteria that are tuberculosis bacteria that using CRISPR, she says she has coated back into their pre-antibiotic state and then used to color the textile here. And her message is more or less with the relation with the controlled commodity campaign of 1941, by which she wants to tell us that we should be careful with applying CRISPR, because you have a lot of antimicrobial resistance where we are losing the effectiveness of antibiotics. And we may lose the same if we use CRISPR technology or technique in an irresponsible way. Yeah, I also wanted to show quickly a few people that are, or a few cases where people are so enthusiastic about CRISPR that it leads to very freaky proposals. On the right, you see a mini pig made by Beijing Genomics Institute in China, which is a pig for doing medical tests that they have made smaller using CRISPR technique. So it's genetically made smaller so that they have more pigs for less space and less food. And they have to kill less pigs for their kind of animal tests. On the left side, you see a Russian biologist that also, after the claim of Yang Qiyui, the Chinese scientist who claims to have 2 CRISPRed babies born that cannot get AIDS anymore. So on the left side, you see this Russian scientist Rebrikov, who wants to do similar and test CRISPR technique on humans, which I think is really breaking the global moratorium on these type of human enhancement. Here's a work that I would love to discuss a little bit more in our discussion afterwards. It's the Chinese-American art and design group, Another Farm, that made this dress-modified paradise that was on show at Ars Electronica, where they genetically modified, and I don't know the exact technique, spider silk worms that now make fluorescent spider silk threads, of which they made this dress that under these light conditions really looks amazing, beautiful. But yeah, I think it's very interesting to talk about the type of modification you make here for making dresses. Yeah, like said, but I'm not going to go into detail here. This is the work of Return to Dilmun of Günther, Roland, and Federico. And the cunning part is that it is not in vivo, but in vitro. I hope the animation works. Yes, it does. Günther yesterday also mentioned this similar piece of work that is also put in motion as an animated GIF. And it was done by the Wyss Institute for Biologically Inspired Engineering, led by George Church. At the same time, or almost the same time, they claim it was 2016, I think very late 2016, and Return to Dilmun was made really early 2016. But this was not done in vitro, but in
Sorry, 17.
Sorry?
I'm sorry to interrupt you. We made it 2017.
Yeah, that's what I mean. So you made it very early 2017, and they made this probably very late 2016. They used an in vivo model, so in E. coli, and not the in vitro protocol that we are taking from you now. There's also, yeah, I would like to make a kind of an overview of the enormous diversity of CRISPR applications in research that is currently happening. Here you see something, there was a CRISPR conference in Wageningen University last year that I joined. And here you see that they are using CRISPR to see whether they can make eels mature earlier so that they can reproduce without the need to travel all the way to the Zaragoza Sea, because they are at the brink of extinction by human consumption. And Wur is really trying to change that nature of this valuable and pretty tasty fish, only for human purposes. You wonder whether fishing more responsibly could be a better method. Yeah, I already touched upon this. This is another speculative work of art, but it also reflects on gene editing techniques. So Huang Yiku, a Taiwanese artist who has been a resident artist at Waag, developed this tiger penis project, where he actually wants to make tissue cultures of tiger penises. And here you see apparently a tiger penis remade in synthetic materials. But if we make a lot of tiger penises in vitro, we don't have to kill them for the Chinese market that sees them as a kind of alternative traditional medicine for heightening potential. Yeah. Also, due to time, I'm going to leave it a little bit here. I could say some more. What we often do is when we do this type of speculative CRISPR workshop with an audience, where we actually do make competent cells with them, where we then stop the whole protocol at the moment where the foreign DNA is introduced into the in vivo model by a heat shock. We start a discussion with the audience about how easy actually the protocol is once you've learned it, but how complex at the same time still the reality is behind it. And we asked them whether they want to do the heat shock and then by doing so violate regulations in the Netherlands, because we taught them so. So mostly people say, okay, let's not do it, because actually we also prep them in a way. But what we do then thereafter is do a workshop on imagining CRISPR applications that they come up with. We make teams and ask them, what would you CRISPR? And often it's very interesting outcomes that appear. So maybe that's also a question for this session. We can ask ourselves what type of genetic modification arts we can imagine. What type of artworks can we imagine that do touch upon the controversies around these techniques that art is so good at in bringing to the public?